Okay, today is Monday, 24. And we're going to talk about Chapter 9, the nervous system. And we are not going to use Chapter 13's figures for these demonstrations. We're going to use Chapter 12's figures, just because this DVD is not booting well. So hopefully we'll be able to do that. So as we get started on this, there's a lot of material in the nervous system, and I was just saying that this is not a course in neurobiology, and it's not a course in anatomy, and yet, we're trying to understand how the tissues of the central nervous system and the peripheral nervous system are set up, and we want to be able to identify them and understand how their cellular structure contributes to their function. So we are on the periphery of a lot of different fields, and so my objective is to give you enough information so that you can understand what's going on, but you're not going to feel like you're neurobiologists after today's class. But this should be enough information. I think one of the things that we've done before that worked well is to have people come up and contribute to a diagram. Remember doing that at the very beginning where we said, okay, there's a cell. And so we drew a cell and a nucleus, and we just started adding organelles to the cell. And it turned out that you guys knew a lot of the organelles that are in the cell. And I think it's the same way with neurobiology. You guys have been exposed to some of this stuff before, right? There's a cell body, there's going to be, these processes, branching processes are coming out. So why don't we come up, just have different people come up, and we'll draw a neuron and identify some of the structures within a neuron. And in that way, we can build from that point. Does that sound good? Okay, so how about drawing the neuron itself? Should I draw the neuron, or does somebody want to come up and draw a neuron? Somebody that hasn't read very much in the book, this is an opportunity to use your art skills rather than what you've learned from the chapter. Is there anybody? Nobody want? Okay, sure. You want to draw us? You want to draw? Yes. Okay? Who thinks that looks like a neuron? Okay. I was going to say it looks like roadkill. Something splattered. No. Or a fish. Have you ever seen that lionfish? Tropical fish? It's got the eye there and it's got these spines. Could be one of those. Okay, so it's not a fish, it's a neuron. All right, so let's start to identify some structures in this neuron. So who wants to come up? Andrea, why don't you come up? You can pick anything you want to. That's part of a neuron. You can choose any color too. Okay. What about the nucleus? Can you tell us something about that? Is it any different, nucleus any different than in a neuron than it is in some other cells? Tend to be larger, smaller, anything particular about it? Shape, round, oval?
Well, it's relatively bigger than this one, and it has the nucleus in it, and much does what a nucleus does in most other cells.
Okay, so you mentioned the nucleolus.
Yes, yes.
Okay, so what's the difference between a nucleus and a nucleolus?
Nucleus is in a nucleus, and it contains the actual information that controls the activities of the cell.
The nucleolus?
No.
The nucleus.
Yes.
Okay. Anybody want to add anything to? Thank you. To the nucleolus, discussion of what the nucleus is and what the nucleolus is. Nucleolus is associated with the nucleus. Doesn't have to be right in the nucleus. It has kind of an orientation function. It's never been completely clarified, but it has to do with microfilaments. Often are associated with the nucleus, and it's when arranging helps to arrange the spatial arrangement of microfilaments through a cell and helps a cell to gain directionality. So one can imagine where nerve cells, neurons, are very sensitive to this positioning thing. They need to know where they are in space relative to each other, and their movement and positioning is a big part of their life. So the nucleolus is going to be an important, it's going to be busy in a neuron. So that's one of the reasons why it has a pretty prominent nucleolus. Okay, that's good. Who else wants to come up? Catherine? It's easy at the beginning, right?
Yeah.
There's a lot of stuff in there, so. No, cell body. You got to give me another name for that. Because biology is all about the synonyms, right? We have all of these terminology. So what's another name for the cell body? You got to stay up here and write it in. Why would they even get into talking about the cell body? You know, where other cells, all cells have a cell body. And then suddenly with neurons, they're talking about the cell body and the cell membranes and these processes that come off. And they give them special names as if this is the first time there's been a cell body. All the other cells have a cell body too. But it ends up being important because they're trying to understand the function of neurons. And so they, going into detail, they want to be able to address what's going on at the membrane, for example, of the neuron and all that. Okay, so somebody else have an idea what the name for that is? It begins with P. It's in the chapter. Harry. No. Yeah. Peripheral. So you write that up there. Peri, P-E-R-I. Yeah, Peri, and then K-A-R-Y-O-N. Great. Okay. Doesn't even need to be called. You knew you were coming up anyway, right? So. Okay, that's a dendrite. What about, is that the only dendrite? How many dendrites? Yeah, a bunch of those. So all those things hanging off the cell body or the perikaryon are dendrites. What about down here? Dendrites? No. Okay, good. Okay, so all of these guys are dendrites. Okay. Good, thanks. Okay, question? did you draw here? What are these guys?
Those are the axons. Are they made of myelin sheath?
Okay, a little bit of confusion there, but okay, that's fine. So you're labeling this whole thing the axon. That's it's a big one. It's a big ticket item.
Yeah.
So good. Okay, dendrites and axons, and a cell body. That's a big part of the whole story here. I thought he was going to talk about these other things he's drawn in the axon. Christian, you're right. That's a more accurate representation of an axon in terms of how it's set up ultimately with some other cell types in there. And I thought you were going to label that, but anyway, we'll keep that. That's a good addition. And this whole thing here is an axon. Okay, Elizabeth. Sure.
Yeah.
Okay, so that would be plural, so it'd be a bunch of Schwann cells, which is what I thought Christian was going to start talking about that. So what do the Schwann cells do? What's going on with that? Well, Christian had talked about this. Christian, what did you say about this? It was the axon had what? Yeah, the myelin sheath, right? So what is the myelin sheath about? Why is that so significant? Yeah, it protects it, but it provides a microenvironment for the whole neuron to be able to function correctly. And the axon needs to be protected. How long are these axons? They can be pretty long. I mean, they give an example of an axon in the foot, and if it's running up to get to this, the spinal cord, it could be a meter long. Yeah, it's like 100 centimeters, is what he said. You know, so I mean, it can go the length of your leg. And so, you know, they're pretty thin, and it's only one cell, so it's got to depend on nutrients and stuff coming from the nucleus, and so, you know, it really needs to be protected. But the other thing about the myelin sheath is what? The whole system, the whole neuronal system is, you know, for us, we're in modern day, we're in an electronic age, so I think this is really poignant to us. What is it about your computer and our cell phones and all that stuff? They work on what? Electricity, right? It's electrical systems. And so they're not just electrical, but they're electronic. Electronics is fine manipulations of electrical systems, complex electrical systems. So, so the neuron is functioning as an electrical system, and the Schwann cells are insulators. So myelin is, here we go again. Let's see, I have these wires in here. So all these electrical wires, they have metal. You guys have taken apart wires before, right? I went through a stage where I took everything apart when I was a kid, including the TV set and radios and everything electrical. And some of the things I eventually put back together, but only a few. Anyway, so the wires have an insulator on them, right? They're covered with rubber or plastic all the time. So the electrical conductor cannot be just hanging out there by itself. It needs to be insulated. And so so that's what's going on in the nervous system. The neurons are sending electrical impulses, and they need to be insulated. And so Schwann cells are great insulators. So we're going to get into that more. Okay. How about John? You can come up here.
Want to say one more thing?
Is it getting tough already or not? Probably not. Okay, give me another synonym before you leave. Axon terminal. So what's at the terminal end? Guess with an S. The thing that it forms with another cell. Okay, synapse. So let's put that down there too, right after the terminal. Synapse. Yeah. Okay, good. We won't pick on Daniel, he's yawning too much. Okay, Catherine, so that's the Axon Hillock, right? Okay, so what is that structure? Why is it a hillock? Is there anything, physically, how does the structure look? Is it thin like this or pyramid shape? Okay, can you make that more pyramid shape? Use the eraser and do an editing of Connor's wonderful thing. Yeah. Okay, a little bit more like that. Yeah, so that's why it deserves this name, Axon Hillock. Thanks. Yeah. So again, I would say that we're starting to get into specialized terms, and why do these cells deserve to have common things get special names? And it's because they're trying so hard to figure out what the heck is going on with these cells, with the electrical impulses and how all this works. And so every little component should get a special name so they can start talking about it and be specific and people will know what they're talking about. Okay, so Dan. Okay. So what happens with these guys? You know anything else about them?
I think that's where the action branches continue.
Yeah. They continue. There's some sort of amplification that can occur there somehow. So that's one of the reasons why it got special attention. It seems to be a natural break in the myelin sheath area, and then there can be some multiplier that can occur. Okay, so how would you pronounce that?
Von I don't know. Von Dier. What's it called?
What do you think, Kai?
Yeah.
Yeah. Okay. It's French. Okay. Let's see the other catheter just opened. Okay. That's a good start. So, how many neurons are there? There's 13 trillion cells. I like that number. I'm always using that number. I'm not sure it's exactly 13 trillion cells in the human body. I've seen estimates up to 30 trillion cells, but I like 13 trillion because it's 1.3 times 10 to the 13. It's easy to remember that. It's 13. But anyway, 13 trillion cells. So there's billions of neurons. So, and it's, in the brain's got a huge concentration of these neurons, right? So that's the main place to find neurons. But the interesting thing is that the support cells for the neurons are more numerous than the neurons. They outnumber them 10 to 1. And the support cells are called what? The general category of cells. And the Schwann cells are one type of sample organs.
Neuroglia?
Yeah, they're glial cells. So we have the neuron here, and we also have this important cell, the glial cell. And the glial cells come in a couple of different variations, but they all play this support role. They're supporting neurons. And there's 10 times as many of them. So that's kind of amazing. So there's, it makes some sense to think a lot about the glial cells too. So, and we will. We'll start talking about the glial cells once we get some more of this going here with the neurons. In the nervous system, there's two main systems. There's the central nervous system and the peripheral nervous system. So the central nervous system is CNS. You've all seen that before, the CNS, the central nervous system. And then what you see much less often is PNS for peripheral nervous system. So the central nervous system is the brain and spinal cord, and the peripheral nervous system is the spine, ganglia, peripheral nerves. One of the things about peripheral nervous system is it's going to be the input for the brain. It's got to be sensory stuff. So it's going to take in messages. So they have a special term for that. They call it afferent. So there's afferent and efferent. So if we diagram the human, so there's the head. We need to have the head pretty prominent. Okay, so we've got this. It's not very good. See, now you know why I didn't draw it. A little better. I'm trying to fix up my human. Okay. Okay, so there's the peripheral nervous system and the central nervous system. So the central nervous system is hanging out in the brain and the spinal cord. So that's central. And the peripheral is everywhere else. So there are nerves everywhere. And that's because you need to be sensing the environment everywhere. And, it's even in the skin here, peripheral areas. So the peripheral nervous system. So I was saying that the sensory, so the peripheral nervous system is all sensory. And this is where the term afferent comes in, where there's signals coming from the outside, and they're going to make their way into the central nervous system. So you prick your finger, there's a little, stimulus here, and then that signal's got to go up the arm, and then it's going to go into the spinal cord, and it's got to make its way into the pain center in the brain. And so this motion, these directional arrows going in this direction, that's where we get into afferent. And then there's the alternative situation where there are things that you decide in your wonderful brain, I'd like to move my arm. And so you make that decision in the cerebellum, and then there's going to be impulses traveling the other way. This is traveling down. So you see the difference in direction, right? So you're giving instructions to a hand to move. I could have drawn it over here, I just didn't want to get the arrows messed up in their directionality. So this is efferent. So far we've talked about the central nervous system, peripheral nervous system, and the fact that when we sense things, there are signals that are going towards the central nervous system from peripheral areas, and that they're referred to as afferent. Okay. What about ganglia? So we've talked about the neuron, and we're thinking about the fact that there's central nervous system, there's lots of neurons in that, the peripheral nervous system. Then there's this other thing, the ganglia. So what is the ganglia? Are those nerves too? Yes, they are. So why are they different? Why are ganglia different than the brain? Well, it's because they're not in the brain. You got to call them something. There's groups of neurons at certain locations, like parts in the spine and stuff. And so those groups of nerves they need a term. So they're anything outside of the brain. But they're also made up of neurons. So they're outside of the brain, they're outside of the central nervous system, outside of the brain and the central nervous system. Okay, at the beginning of the chapter, they talk about a lot of this terminology, but they're not talking about cells exactly, but I still think it's worth going through this stuff because it orients you as far as what is going on with the nervous system. So we should go through it. Stimulus response. That's common stuff if you took neurobiology. Stimulus response. So what do you think that's about? Stimulus, if you prick your finger, that's a stimulus, and then you're going to have a response. You're going to pull your hand back. Ouch. Right? Stimulus response. That's on a gross level. But, on a more cellular level, if the cell is perceiving something, it's going to respond in some way. So, let's discuss just the cellular level of this. So we mentioned stimulus and response. Red. So on the gross physiological level, you pull your hand back if your finger was hurt. But on a cellular level, there's a stimulus and a response. How is this neuron sending and receiving messages? Well, it's through this setup that you guys have all drawn. And one of the key components here is this synapse, and the other key component is the dendrite. So at one end, there's a potential to take in signals, and at the other end, there's potential to transmit those signals. So again, how does this work? Well, there's an electrical potential. And that's kind of it's a very, if you take the beginning of electricity class, they talk about voltage potential. That voltage is a potential. It's a so to call it an electrical potential is to say that that you really are dealing with kind of an electrical system, but it's not wires like what we're used to with all these things. What you can get, you can transmit electrical signals by moving around ions, and that's the amazing invention here. And I think it's pretty impressive that biological systems somehow took advantage of ion flux across a membrane. So you guys took some basic chemistry where they talked about the membrane, like a semi-permeable membrane, right? And we were saying when we talked at the beginning about cells that they're covered with a plasma membrane, and it's a semi-permeable membrane. So if you take a cell and you plop it into water, what happens? Does water go in or out, or what happens? You take a cell and you put it into water.
It'll explode.
It will explode. Why is that? So it will explode. So that must mean that water is going in. Yeah, it's rushing in. So why is the water going to rush in?
water wants to go into a concentration of sodium or high salinity?
Yeah, so there's higher salt concentration within a cell, is what you're saying. So there's sodium in there. Another example would be potassium. There's a variety of ions. There's some calcium. There's many more ions as well. But water is going to go in across the semi-permeable membrane that try to dilute out where there's a higher salt concentration. So what happens if you take some cells and you have a concentrated salt solution and you drop them into that nitrar? What would happen? Cells go into a more concentrated salt solution. You just mixed up some anchovies with sea salt and it's real salty, and some cells get in there. What are they going to do? Are they going to explode like John said when they went into water, or is there going to be a different outcome? You think they'll mix in better? Okay. Well, that's a good thing, firstly, right? So they don't explode. So that's a plus. So that must mean that there's ions on the outside and there's ions on the inside of the cell too. So that's a little less jarring, right? Is perhaps what you're thinking, right? Okay, so that's good. But let's say there's even higher salt outside the cell dressing you make is even higher salt concentration than inside the cell. So then the cells are going to do what, Connor? They're going to what? Crenate?
It's called crenation.
Crenation. Cool. Yeah, they're going to shrivel up, right? And so the water is going to come. It's going to do what? It's rushing out. So that's just a semi-permeable membrane. It's simple, like a dialysis membrane. It has certain pore size, and certain size molecules can go across that membrane. And so, water is a pretty small molecule, right? H2O. It's not too big. What's the molecular weight of oxygen is 16, and hydrogens are 1, so you got a molecular weight of 18. No, it's a small polar molecule, water. So it's going across the membranes. Ions are pretty small. Ions are small because they're not connected to other atoms. So they're not making larger molecules, they're just single ions. They're single elements. Everybody know that stuff is basic chemistry stuff. I'm just trying to remind you of this because you need to think this way to understand the action potential inside of a cell. So it's electrical potential, and they sometimes call it action potential as well. And as I was complaining with biology, you need to know all the synonyms because you're going to get confused if you don't, understand what those are. And a lot of times people are talking about the same things but using slightly different terminology. Okay, so water can come across this membrane, but ions, well, the molecular weight of calcium, well, let's see, sodium is what? 35? Is that? Now, I'm not sure. Calcium, let's see, chlorine might be 35. Is there a molecular weight table? Anyway, so these things are small, right? So they're single atoms, and even if they have a higher molecular weight, these pores are still way bigger than any kind of single atom, even ones with high molecular weights. Makes sense, right? Okay, so the ions can go through that membrane, just like we said. You take a cell and you plop it into water. Well, the water comes rushing in, so water can go in. But if you have salt and you get crenation, so you get crenation to occur, and then the salt is going to go. The water is going to come rushing out, and the salt's going to end up going in. So ions can go across these membranes, and the main ions involved are sodium and potassium. So let's draw those guys. So there's this thing called the Sodium-Potassium Pump, which is driven by ATP. And it keeps the inside of the cell, the sodium level, sodium is kept low, So inside of the cell, sodium is kept low, but calcium is kept higher than the outside. Low, high versus outside. Right? So that's one of the things that's crucial to a neuron, is that it has this sodium-potassium pump, and it's driven by ATP. What does ATP do?
Energy.
It's what?
Energy.
It's energy. It's the energy currency of the cell. Just looks like our dollar. We're trading everything in dollars. Well, the cell is doing all sorts of things, and it's using ATP all the time to get it done. So it's linked to the overall energy of the cell. So the cell expends some energy to keep the sodium concentration lower inside the cell, but the potassium level to be higher. Because this is low, the sodium level is low, if you want to transmit a signal, you can have sodium come rushing in. And if it will come rushing in because it's kept low inside the cell. This is lower than physiological concentration. And so there's a potential difference there of some number of millivolts. Like, here we go. Electrical potential difference of that axolemma is and there's minus 65 millivolts. So there really is something electrical that you can measure with these cells. And if you go to a neurobiology lab, they actually have very sensitive electrical machinery with probes and things, and they can see electrical changes occurring in the cells as the neurons are stimulated. They can watch to see the action potential. You ever seen that? They have like an oscilloscope connected, and they can, with a trace of the oscilloscope, they can see a action potential going down a neuron. So it really is something measurable and electrical. And I'm struggling here to try to explain to you guys how ions going across a semi-permeable membrane are equivalent to electrons going down a metal wire. Because our whole world is metal wires with electrons right now. And you guys are looking at computer screens and stuff. You're manipulating those electrons in very careful ways. And our body is also regulating electrical potentials in very careful ways too, but it's not just shooting electrons down, it's using ions. And so the ions have to go across this membrane. How quick are the ions? Well, milliseconds. So that's pretty quick. Millisecond is how much of a second? Yeah, it's 10 to the minus third, right? So it's a 1/1000th of a second. So a second is already pretty quick, but our bodies are responding in milliseconds. So that seems good enough for the real world. We're probably quick enough. If we were hardwired, it might go even quicker, but milliseconds is pretty good. So does that make sense to everybody that if you stimulate a neuron, and so if a dendrite is taking in a signal and you want to conduct an electrical impulse down a cell, that you can have a momentary flux of sodium across this semi-permeable membrane, which is the axolemma, so they give it a special name. It's just a plasma membrane, but this is a neuron. So, and scientists spend a lot of time playing with neurons and trying to understand neurons, so they give it a special name, axolemma. So you can conduct an nerve impulse just by having a wave of conduction of sodium ions rushing in, and then you're gonna stop them from rushing in. So it's a wave that occurs. It starts out up here, and then it's going to go down the axon. But as soon as it occurs, as soon as the sodium ions are brought in, they're going to be pumped back out, right away. Because all this stuff happens within milliseconds. So it's hard to fathom, but it's also hard to fathom why electrons can go shooting down the wire so quickly, right? And it's hard to fathom why your computers work so wonderfully with all the different electrical connections that are in there. So our bodies are pretty much analogous to that. We're just using a slightly different system. So we're not just using electrons, we're using ions. And the ions are going across the membrane and they're conducted in a wave. And so when it takes in the original impulse at a dendrite, there will be a flux of sodium ions that will come in, and then it will move. That flux will move down all the way down the axon until it gets to this terminal area, the synapse. What happens then? It's the same game or a different game? So the game of electrical potential here, how a neuron is communicating a signal. We said it's all about this sodium-potassium pump, ATP driven, and that flux of ions across the membrane, across the axonlemma. And when you come to the end, is it going to be another ion flux thing at the synapse? No. The game changes here. There's a different signal. So when the axon is, so we draw another cell. So this axon is touching a cell body of some other cell. Could be a muscle cell, could be another neuron. And the synapse is this region between the axonal button or bouton and this. There's a synaptic cleft here, so there's all sorts of terminology around this, but there's a region where this terminal region of the axon, it's going to transmit a signal. And we're saying that that's not electrical. So all the rest of it is electrical, but here there is a transmitter. So a neurotransmitter. So that's kind of interesting. So a big part of the system, it's all about electricity, but then suddenly you come to these ends, and then you have neurotransmitters instead. And there's a bunch of different molecules that are neurotransmitters, but I'm not going to talk about that yet. Okay, so back to cells. I just wanted to give you, a feeling for where we're going with this, because we're talking about the structure of cells and the function. That's what this class is all about. The structure, what do cells look like? What's their structure? How do they accomplish the job? So we have the basic story here. We're dealing with the nervous system. We drew a neuron, talked about an electrical potential, and then it's going to interact with the second cell and use neurotransmitter to continue the transmission of that original impulse. But every time you create something different, every time you have a connection point, there's an opportunity there to vary the signal, to change the way that signal is going to be interpreted, and that's really important because in your computer's doing that. Your computer has a microprocessor, and the microprocessor is taking in lots of bits of information, and it's processing those in some way that makes sense. A lot of it is just arithmetic, and that's the way the neural system works too. There's a summation effect. Neurons can sense when other neurons are signaling, and they kind of look around at all the connections that they've got and how many signals are coming in and what's the appropriate response when there's a fair number of signals coming in, or let's say there's far fewer signals coming in. It would affect how the signal is going to be propagated. So it's actually a strength of the system that there are so many neurons involved with transmitting a signal. It gives you opportunity to amplify, to have more, to bring up the signal strength. It gives you an opportunity to lower it, and it gives you a choice on which direction you want to send the signal. Okay, so boy, there's a lot of material here. There's a diagram here on the development of the nervous system, and I think it's okay in this case to look at that because it is such a unique system. So let's I hate to do it, but let's erase this part. I think we got a good start on it though there with that diagram. Actually, we could see if this comes up on the DVD, I'm not sure. Yeah, the third edition that we're working with is quite a bit revised. There are some definite changes in the text, and I think it's better. So I'm glad we decided to go with the 13th edition. Okay, so I'm going to have to draw this. So you see it in your book, but I think it's worth, this should be something that's in your mind too, in your understanding of what's going on with neuronal systems, because it gives you an idea of how the whole thing works together as a biological unit. So this is a notochord, and this is a neural groove. And then there's, let's see, this is going to fold up. So what's significant is that that from a situation where there is an area where there's ectoderm and the developing embryo has an area of neural cell types, there's a fold here, these neural folds. And basically this is going to curl up. You see this in the book, right? It folds up like this. And once these two ends meet, you've basically formed your the whole backbone. So this is crucial to creating a central nervous system. But anyway, the groove, the neural groove, and it folds up, it grows out, it folds up, it connects to the other side, and then you've got this tube thing here. And just above the tube region, as this folds up, there's this other set of cells that are just above that, and that is the neural crest. Okay, so we're trying to make this transition, right? We talked about this, just setup of the cell, and then a little bit about how this impulse is transmitted, and then now we're getting back into the cells and talking about, well, how can we understand what the cells look like, what their structure function is, that they are able to accomplish this job. And so there are cell areas that are associated with the neuronal function, but there's also neural crest cells which are going to participate in the neurological system, but they give rise to mesenchymal cells. And these mesenchymal cells are, they form a lot of the glial cells. So we're put down the glia up here, glial cells. So they're derived from neural crest, which is more of a mesenchymal cell type here. So we're histologists. So for us, that triggers a certain ideas about mesenchymal cells. Those are the ones that form what? Give me some synonyms for mesenchymal cells, and then what do they take away? Yeah. John? Mesenchymal cells.
It sounds familiar, but I can't remember.
So, Connor said stroma. So, another term for stroma. Not sure. What? Yeah, exactly. Good. Connective tissue, right? We were very, very comfortable with epithelia mesenchyme. I hope we were comfortable with it. I was comfortable with it. I've been studying that for a long time. So the body is set up where there's usually an outer layer. Remember we were saying that epithelial cells are the outer layer cells, and then the support cells that they interact with are mesenchyme. And the mesenchyme is the same thing as connective tissue or stroma. There's not that many cells there, but there's a lot of extracellular matrix, lots of collagen, lots, there's some elastin and other sorts of filaments. And so that was a very typical setup. Here in the nervous system, it's, we've got some cell types that we can start to relate to here. The neural crest gives rise to mesenchymal cells. Are they going to create that kind of same scenario with the connective tissue? And the answer is no, not really. What ends up happening is that the glial cells, there's a tangle of all sorts of processes from neurons and from glial cells, glial support cells. So if you look at a picture of this in the text, you end up with something that looks like connective tissue. It's figure 98, and the 12th edition doesn't have this figure that I can see. But if you look at it, yeah, they don't have that figure. Let's see. So that kind of looks like connective tissue, right? It's a web of stuff. And you see a couple of cell bodies in there, but you see a lot of strands and things. So one thing that you could guess, if I showed you a picture of this, you might say, well, that looks like connective tissue. But then they stained it with gold chloride over here. And the gold chloride does a good job at staining all those fine structures of neurons. And so then it doesn't really look so much like connective tissue, right? Because connective tissue is you've got collagen fibers, filaments, and you've got these other extracellular matrix proteins, but those are very fine, right? You're dealing with molecules there. Here, this isn't a molecule. These are strands of the cell. These are axons and dendrites, and there's such tangle of these things that they take up the space, and it looks a lot like connective tissue, but it's not. And so there's not really much collagen in there, and there's not a lot of the other extracellular matrix proteins that we're used to in there. It's its own deal. So that's the one thing about the whole nervous system that's quite a bit different than everything else that we've discussed up to now, is that what we've become so comfortable with in epithelial mesenchyme doesn't really hold up very much. There's not really many examples of epithelia either. There's like an epithelial-like covering that's shuttling around the cerebral spinal fluid. But neurons are their own group. And for some reason, the neurons like to stay with the heads, the bodies of the cells like to hang out in groups, and the axons that are myelinated sort of hang out in areas too. And if you look at a slice of the brain, you see gray matter and white matter. and the difference between the gray matter and the white matter is just the amount of myelin. Where it's white, there's a lot of fat, there's a lot of lipid. So myelination is extra lipid. And so the white portions of the spinal cord and the brain are these myelinated sheath-covered axons, and the perikaryon, the cell bodies are concentrated more in this gray matter area. And so I'm just trying to give you an overall difference there. You're looking at something different. You can appreciate that, right? You know, when you think about a slice of a brain, it looks a lot different than a slice of everything else. You know, also a slice of the spinal cord. You've got this matter in there, and there's areas of white and areas of gray. And so it just has to do with how many cell bodies there are in there and how much myelinated sheath there is on that. Okay, so although this is mesenchyme, you know, it's a different deal. Glial cells are support cells, but they're not extracellular matrix factories like fibroblasts were. They're support cells, and they can be intimately involved with the neuron, and we showed that in the diagram. I guess Christian came up here and he was drawing the squares around the axon, and then you know, what are those squares that he drew? Those are Schwann cells, and they actually multiply wrap around the axon. It's really amazing. You know, if you look at a cross section of that, you see it looks like it's wrapped several fold around. It's like if this is an axon and I started wrapping paper around it and I just kept going, wrapping and wrapping and wrapping, you know, I'd have a couple of layers full. And so the myelinated axons are like that. They're really well insulated, and the glial cells are doing a lot there. So in addition to just insulating it electrically, they're helping to sustain the balance of ions. They're keeping things localized. They also provide physical support for the neuron. So there's a lot that they're accomplishing. Okay. So we're kind of bouncing around here, but I just, you know, wanted to give you an idea of this. It's a different structure, but it's very specific. During development, there are dedicated cells that are going to become the nervous system, and everything else is not the nervous system, right? And the neural crest is giving rise to these glial cells that are support cells, but the neural groove, and then ultimately the neural groove gives rise to the neural tube. So once it's a groove, and then once it closes up, it's a tube. Neural tube. And that's the primitive spinal cord. Okay, they distinguish a bunch of different arrangements of classes of neurons, they call them. And it's just an example, I think, where you can see that there's different ways that neurons can look. So the categories they've come up with, classes, there's a multipolar neuron, there's a bipolar neuron, a unipolar neuron, and an axonic neuron. So the first one, multipolar, is going to be a lot of dendrites, kind of the way Connor drew our neuron. So multipolar, there's lots of possible input into the neuron and then transmission down through the axon. In a bipolar neuron, the amount of branches of the dendrite are very few, and there's just one process coming out of the cell body, so it looks kind of similar to the axon. The axon, remember we drew that axon just as one long thing. In general, that was the right diagram to have in your mind. Axons are the main process coming out of the cell, that's going to the synapse. And the dendrites, there are multiple groups of them up at the top, but they can look like an axon on the other side. And then you would call that bipolar because one side kind of looks, the dendrite side looks the same as the axon side. So that's why it gets that name. And actually, the bipolar neurons are used in sensory organs, things like the retina and olfactory mucosa and the inner ear. So your sense of sight, smell, balance, those things are being transmitted by this bipolar neuron setup. And unipolar are similar setup, but there's only one Axon Hillock type of looking thing, and the dendrites and the axon is coming both out of just one location. So that's why it's unipolar, one site. And axonic means that there doesn't seem to be any axon. You've got the cell body and then these bunch of dendrites hanging around. Okay, so why would they categorize things? They're trying to understand how the nervous system works. And so any observations that can be made to help, categorize things and give us some bearing on the way these impulses could be processed is helpful. I mean, clearly, I think neurobiology is a work in progress. And I always felt that it was running a bit behind other cell biology, but it's probably because the inherent systems are so complex. So I always thought that it's kind of, given the current technology, it was kind of rewarding to work on non-neural system cells. But as we get better, the neuronal systems are very appealing, and possibility for enhancing somehow the human condition using some of that is kind of appealing. Okay, so dendrites. So dendrites have these additional connecting points, and they're just a way of giving you lots more connecting points. So if there's a dendrite that's coming out, then each one of these dendrites has a bunch of little protrusions on them, and these are called dendritic spines. So these are possible points of connection and transmission of a neuronal impulse. Dendritic spine. So you can see where, rather than just using one dendrite, one branch of this tree-looking thing as being a connecting point, each one of these dendritic spines could actually be connecting to something from another cell that's transmitting a signal to them. So that's a way of multiplying things. So dendritic spines help to explain why explains that you've got hundreds of connections. Hundreds of connections to just one neuron. Just to kind of try to understand this game. So it's not a matter of just, the wire we had is sort of a simple connection, but the neuron is taking in lots of different signals and hundreds of these signals, and they can connect through these dendritic spines. One of the parts that I like about this is I like talking about the some of these neurotransmitters and stuff. Let's see. Most synapses occur on dendritic spines. There's about 10 to the 14th for cells in the human cerebral cortex. Wow, it's more than hundreds. But anyway, so that's a number that they gave in the book. I'm not sure what they're talking about there exactly, actually. but I guess maybe the sum total of all the connections, all the synapses through dendritic spines are 10 to the 14. So it gives you an idea of how many, there are billions of neurons, and there would be hundreds of billions. So I can't really get up to 10 to 14. Anyway, so those are initial processing sites for synaptic signals. This Purkinje cell, the cerebellum, is a very large neuronal cell. That's an example of a cell that would have lots of potential dendritic connections. So it shows you how interconnected these guys are. Okay. There's terminology like terminal arborization. You heard the word arborization or an arbor? What is an arbor? I knew a company called, what was it? The Tree of Life, Arbor Vita. Arbor Vita is a biotech company locally, and so they use the word arbor tree is Latin for, arbor is tree in Latin. And so arborization means that this is branching, extensive branching at the ends of axons. There's this term collaterals, which are branched ends of synapses that influence other neurons. And their ends are called the terminal bouton. So collaterals are branches at the ends of these synapses. So anyway, we get the picture that there's a lot going on with the connectiveness of neurons. And so that's one of the things that you can see when you look at sections of neurons. You can try to call up some of these pictures from the second edition, see what they look like. Here's a motor neuron. It's a large cell body. Contains a large number of Nissl bodies. Anybody pick up what a Nissl body was? So one of the things about histology is it's a classical study which involved the use of chemical stains to stain tissue slices, and we had this tool, this new tool, the microscope, and we could look at these chemically stained sections. And then we saw that the chemical stains were taken up differentially in different types of cells. And then the cells, as they were responding different ways, would sometimes stain differently as well. So we get an idea of the types of tissues based on their chemical uptake. So a Nissl body turns out to be just a bunch of rough endoplasmic reticulum. So if there's a lot of rough ER inside of a cell, then that gives you a kind of a purplish stain in some of the staining protocols, and so they called that Nissl bodies. Probably there was some researcher named Nissl that saw this. So he was correlating that the graininess in the cytoplasm of a particular neuron to something about its properties. So that's just a way of saying, well, neurons that have more of a need for a rough endoplasmic reticulum are going to be a unique population compared to those that have less of a need of a rough endoplasmic reticulum. What happens in the endoplasmic reticulum? Again, do you remember? So proteins are made on ribosomes. And ribosomes look like little spots in cells, really, really small. And ribosomes are made out of ribosomal RNA, the two different sizes of ribosomal RNA, and then there's associated proteins, and that makes up this protein translational machinery. So there's free ribosomes, polyribosomes, and then those are ones where if there's messenger RNA floating around, these ribosomes can bind to the initiation site and start translating a messenger RNA into protein. But the protein isn't going to go anywhere. Once it's translated, it's whatever that signal said it should be is what it's going to be. It's going to be a linking, beads on a chain of amino acids that make up a polypeptide, and then if it's bigger, a protein. Right? So that's what a free polyribosome is. But rough endoplasmic reticulum is associated with this internal membrane inside of the cytoplasm. So, the Golgi apparatus was part of that too. Remember when we were first talking about cells, we said, well, there's kind of this internal membrane. Not only is there plasma membrane, but there's internal membranes inside the cell, and they're all contiguous with each other. And when you have a lot of the ribosomes translating protein right on a membrane, then it needs to go through the membrane, and that's where you start using signal sequences, saying, well, okay, we're going to allow you to go through certain membranes if you have certain amino acids at the end of the protein. It's going to be a signal sequence, so then it's going to be able to move into the Golgi, and then in the Golgi it can be modified with post-translational modifications. We can add sugar groups to that, and so it's basically all protein synthesis machinery. Why would a neuron need all of that protein synthesis stuff? Well, neurons, for one thing, they support a long axon, right? And they have this nerve potential thing. We were saying that a neuron could be as long as your leg, right? Going from your foot to the tip of your toe all the way up to the spinal cord. So there's that cell body has got a big responsibility to keep that whole thing fed. And so it's going to need more synthetic machinery. It's also going to need synthetic machinery to maintain those Sodium-Potassium pumps. It's going to need to make neurotransmitters that operate at the synapse. The more active, the larger the neuronal cell is, and the more active it is, it's going to need more of this rough endoplasmic reticulum, and so that ends up being this Nissl body observation. So, I mean, it's just interesting for us. It doesn't really get at how neurons work, but this is not a neurobiology class. This is histology. So, so when you look at cells, this cell here, this has a great number of Nissl bodies. So it's kind of it's heavily stained in the cytoplasm, and and then they just talk about some of the other things. But they did point out the nucleolus, and we said that the nucleolus plays a more prominent role in the nervous system than it does in some of the other systems, because neurons really need to be oriented correctly. And the nucleolus has a lot to do with microfilament orientation and overall the orientation, directionality, and polarity of the cell. Okay, when looking at the synapse, they showed some very high-resolution pictures. What kind of picture could you take of a synapse? Would you need a special microscope? You would. Why? Small. It's really small, right? So a neuron is a single cell, but the axon is just part of the cell, and this terminal bouton at the end, the neural synapse, is just a small piece of a cell. So to really see what's going on in a synapse, you would really need super high magnification. What kind of microscope, Catherine? What kind of microscope would we use to see the synaptic vesicles inside of the terminal bouton of the synapse axon? A what? Electron microscope, good. And what kind of electron microscope? SEM or TEM? Right, TEM. So transmission electron microscope. So those are the microscopes where you can take a section of what you're looking at and you can see the internal workings of a cell or even part of a cell. And you can visualize the synaptic vesicles. I don't know, if you look at figure 9.6, they have a TEM there of synaptic vesicles. And you see the areas where the synapse is interacting with other cell types. And these vesicles are being exchanging neurotransmitters. So the neurotransmitters, they're a way of communicating the fact that there's an electrical impulse going down one neuron and it's going into another neuron, and transmitters are used rather than just continuing on with the electrical impulse. Why is that? Well, it turns out to be useful, right? Just like the microprocessor of your computer has a lot of input coming into it, and a lot of different bits of information, and every time there's an exchange of information, some interpretation of that information gives you higher brain function. So the use of neurotransmitters is a way of getting, interpreting the data, interpreting just electrical impulses and saying, we've got a lot of bits here of a lot of tingles, a lot of things, electrical impulses coming in, and I sense, I've got 100 different connections going through my dendritic spines, and I see a lot of signals coming in. What's my interpretation of this? What situation is this? And it can affect what kind of release you're going to have into the synapse. And also it depends on the type of neuron and what would be expected. Okay, so neurotransmitters, though, they must interact with the overall system, and we were saying that the electrical impulses are carried by ion channels. Opening and closing ion channels is the way that you're getting an electrical impulse to work. So the neurotransmitters must have an effect on that system too. They're intermediates. So once the synaptic vesicles are release some neurotransmitter into the synaptic cleft, the other cell type is going to respond to that, but it's going to respond by, again, opening and closing ion channels. So you end up with going between these two systems of electrical impulse versus neurotransmitters. There are neurotransmitters that are excitatory in excitatory synapses and ones that are in inhibitory synapses. So they can change the concentration of ions present. The excitatory open up more of the sodium channels, and the inhibitory cause some of the chlorine channels, the anions concentration to change. So these neurotransmitters can cause hyperpolarization, and they could make a membrane more negatively charged, for example, which would make it more resistant to depolarization. If you change the balance of ions inside the axon as it's going to be transmitting signal, then you'll have an effect overall. And how sensitive are we to electrical impulses, and how much stimulation do we need in order to propagate the signal? So you can have excitatory synapse and inhibitory synapse. The impulses are modified at the synapse by similar connections to other neurons as well. So there's an interpretation aspect to this. So they mentioned that the neurons are determined, this response is determined by a summation of activity. So let's write that down. So multiple impulses are coming in, and then those have to be interpreted. And then as those are interpreted, there'll be appropriate neurotransmitter response. And this is where the interpretation is what they refer to as a summation. So I think this is I know it's a little painful, but it's a key concept that every time the neurons are interacting with each other, there's a chance to interpret what's going on, and that adds to the value overall of the electrical impulses. Otherwise, they're just a bunch of bits flying around like in your computer. But when there's a microprocessor, the microprocessor is interpreting the bits that are coming in and acting on that and giving you some kind of predictable response. Well, the neurons are like that. There's a lot of impulses coming in, and then they get a chance to interpret what's going on. And part of that they call summation, and probably, actually in some cases, probably is just a summation. You know, how many impulses are coming in, and if all the panel is lighting up, that must mean that we have to take drastic action. So we're going to have an appropriate neurotransmitter response to tell a muscle cell, like for example, this neuronal cell might be interacting directly with a muscle cell. And we say, okay, contract like heck to get your hand out of there because you've just stuck your finger in fire, and so you need to respond quickly. So these neurotransmitters, though, the neurotransmitter, it's chemical, some kind of chemical. Whereas before, what we were talking about was in the propagation of a neuronal signal, we were talking about ions, right? So the first part of the neuronal signal is ion mediated, but the neurotransmitter part is chemically mediated. So chemicals they're more complex. An ion is sort of some chemical, but it's just an element, right? The sodium or potassium ions. But the chemical neurotransmitters are more complex. So if these are chemicals, in some cases they can be amino acid derived. Amino acids or amino acid derivatives, and they can be peptides, neuropeptides. So what's a peptide again versus a protein?
The string of amino acids.
Yeah, peptide is a string of amino acids. Peptides are typically smaller. If you have 20 amino acids, that's fairly nice sized peptide. But proteins are longer than that. They can be hundreds of amino acids long, and then you get a secondary structure where you get this folding and cysteine bonds are forming, and you have a tertiary structure to the protein, and it has a shape, might have enzymatic activity and all that kind of thing. But peptides are often signaling molecules, and they're just a couple of amino acids long, and they tell cells what's going on. So those are examples of neurotransmitters, and they're chemically derived. So if at the end of the neuron we have the synapse here, so this is the terminal button, synaptic cleft. We have the synapse here. We've released neurotransmitters into the cleft, and these things are things like amino acids and peptides. Well, you could break them down. I think the ion system seemed a lot cleaner, doesn't it? You're just having this short millisecond flux of ions across the semi-permeable membrane. You transmit that down the length of the cell. That's all good. You come to this junction point, though, where you've got a synapse, and then you're going to start using neurotransmitters. So it's a chemical. So the chemical is not the same as an ion, and you have to once you use a neurotransmitter, you have to get rid of the neurotransmitter. That's the problem with it. That's what I'm trying to get at. So if you're gonna signal something on, you have to take away that stimulus, that it's off. Otherwise, it's meaningless. Right? Does that make sense? So an effective signal has got to be one that you can say, hey, I lift my hand and you respond to that. Oh yeah, he just gave us a signal, start the race or whatever. The hand went up. But then the hand's got to go down. I mean, if the hand was always up, then there wouldn't be a new situation, right? You wouldn't know. Yeah, start the race. Well, your hand's been up the whole time. Now what? So neurotransmitters need to be taken away, and in order to get rid of them, there's a couple things you can do. You can have enzymatic breakdown. So why enzymatic breakdown? Well, peptides, right? Those could be digested up. You can break down larger chemicals enzymatically. So enzymatic breakdown is one way to do it. You can have just diffusion. What does diffusion mean? Diffusion means that there's some local concentration of some chemical that was released and it just passively diffuses into the surrounding area. It gets diluted out kind of passively. So that's one of the things that can happen to a neurotransmitter at the synapse. It could just diffuse away. You could also get endocytosis. Of the neurotransmitter, and that means it's going to be recycled. It's going to be endocytosis is where the cell can, in the membrane it can wrap around some molecule and form some little vesicle, and then pull that vesicle in and digest the contents there and recycle the material. So endocytosis is a sort of a recycling effort. I know this is long, but I think it's a complicated chapter, and so I think it's useful to just talk about this stuff, and you can look at it at your leisure. But it is important to have some background in neurobiology in order to interpret what the slides are showing us and knowing what the differences are of different neural systems. So an axon that synapses with the cell body is called axosomatic. Right? Somatic. So axo for axon, and then somatic cell body. And then if the axon connects to a dendrite, it's going to be axodendritic. So axon synapse to dendrites. Then you could have the axon connect to another axon. So then that would be axonic. Axon synapses to another, different axon. So axons can connect to a variety of different other cell types. And, as people interested in histology, you can see examples of that. And so the picture might show you that the neuron is interacting either with a cell body. It looks like the terminal with Tom is connecting right to a cell body, so that would be an axosomatic connection. If it looks like it's connecting to a dendrite, axodendritic, or to another axon of a different cell, axoaxonic. To see dendrites, well, the gold was useful. That gold staining, gold chloride. You remember what that one looked like? So, also, silver precipitate seems to be good too. That some of the pictures where they were able to see dendritic spines, they use silver. So gold and silver are heavy metals that are useful for pointing out really fine structures. That's why they're used. Okay, so we've got these axon connections. What are these neurotransmitters? I think this is interesting, although it's not, it's not the cells. It's not looking at cells, but we should at least cover this, I think. So we have things like acetylcholine. And that's it used at neuromuscular junctions. You also have catecholamines. And an example of that is epinephrine or adrenaline. How many people here have heard of epinephrine before? How many people have heard of adrenaline? How many people knew that adrenaline and epinephrine are exactly the same thing? Yeah. Okay. So it's just kind of funny, again, where the synonyms, all the synonyms in biology. So other examples, catecholamines, norepinephrine, dopamine, etc. Okay. Amino acids. We had written that down before. Amino acids, and that turns out to be a big category. Glutamine. Glycine, which is not an amino acid, but it's on the list. Well, but a big one here is serotonin. Who here has heard of serotonin before? Yeah. Where have you heard about it? Have you heard about it in terms of drugs at all? Seems like one of the things that. No? Where did you hear about it?
Well, I used to sell this machine and claim that it would increase serotonin.
But you don't believe it?
Nah.
You were selling a machine, you didn't believe it?
Yeah.
So did you feel bad about that?
Yeah.
So you decided to go back to school and get a real medical education.
Oh, yeah, I didn't.
So how did that machine affect serotonin?
Whole body vibration. You seen it all.
Was it a chair?
No, it's a little. You stand on it. Yeah, it's a platform and it vibrates. It's supposed to increase blood circulation and everything.
Was it expensive?
Yeah. The machine was only like $500 to make, but it was selling for $2,000.
Okay. Well, that's not so bad. A lot of things you buy are you're paying multiples. Oh, you're always paying multiples of what it costs to produce. So it cost $500 to make, and you sold them for $2,000.
Yeah. Like $2,000.
So what was the proof then? I mean, why did they pick on serotonin?
I don't know. They just gave me a book to read. And they said this and
Yeah.
said it.
Well, the interesting thing about serotonin is that it's linked to more complex behaviors. To feeling good. So they like that idea. They're not going to say, well, this vibrating platform does something to the Sodium-Potassium channel, ATP-driven sodium-potassium channels. think most customers would say, oh, that doesn't sound so important. But serotonin is actually a target for a lot of drugs. There's anti-anxiety drugs. And, I mean, we're a very heavily medicated society, so it's constantly put out there that we need to be medicated. Some of it probably is pretty useful because I guess our systems are not perfectly in balance, and just perhaps naturally there's some variation in how many people are perfectly balanced. Probably environmental factors are messing us up a little bit, maybe. Some exposure to insecticides and things, maybe affecting our neural systems. But anyway, so amino acids, serotonin is in that group. And the chemical name for serotonin is 5-hydroxytryptophan. Or tryptamine, sorry. And short for that is 5-HT. So this kind of stuff is very popular on MCAT exams. The whole neurobiology thing, and you're not going to see any pictures at all. So, most likely anyway. That's one of the reasons why I think it's worth getting into more of the mechanisms in neurobiology than I want to. The thing that's dissatisfying to me about it is that the nervous system still isn't really understood. It's not really understood how we store our memories and things that I would want to know about. If you're gonna, if I'm gonna learn all this detail, then I want to know the punchline. I mean, how is it that I remember something? And they don't really know that yet. But what is a big thing though is that these neurotransmitters are targets for drugs, and we're all heavily medicated. I mean, the pharmaceutical industry wants to sell anti-anxiety medications, and if you have attention deficit disorders, or overly aggressive, or if you want to curtail your appetite, if you're eating too much, or if you're not able to concentrate and you want to improve cognitive ability, learning, memory, if you're kind of down and you want to be a little bit more up, you could increase serotonin levels. So there's quite a bit going on with serotonin. So what is that? So we're saying it's an amino acid. What's the structure of amino acids? Somebody come up and draw me a basic amino acid, just like what an amino acid looks like. Connor, you don't remember? And I'm going to med school. Come on. Think. Basic structure. So there's an N and C terminal N. Remember that? There's something in the middle. It's going to be some kind of carbon. There's a side group. There's going to be a C terminal N, which is C, and then COOH. And there's an N terminal N. So nitrogen or an amine, and the nitrogen has 3 hydrogens hanging off of it. So that's the basic structure of an amino acid, right? So we just change the R group, and then you get a different amino acid every time. So where does serotonin fit into this? Well, 5-hydroxytryptamine. So tryptamine comes from tryptophan. So tryptophan. So what happens is that when you make these amino acid derivatives for neurotransmitters, you chop off some of it. You chop off the C terminal N. So if you chop this off, and then you make use of this R group here is going to be tryptophan derived. So tryptophan has a ring structure. And I'm not sure this is going to be 100% accurate, because I'm just trying to draw this on the fly here, but